Welcome back everyone. Today we are going to put that knowledge into practice. We'll be walking through 5 practice questions from the hematology session. Okay, question number 1. Hypersplenicity is characterized by A. Polycythemia, B. Pancytosis, C. Leukopenia, D. Myelodysplasia. The correct answer in here is C. Leukopenia. What is hypersplenicity? Hypersplenicity is a condition where the spleen becomes overactive. One of the spleen's main job is to remove old and damaged blood cells from circulation. When it's overactive, it removes too many cells, leading to decrease in one or more cell lines in the peripheral blood. This is called cytopenia. The first one, polycythemia, is an increase in red blood cells. That is the opposite of what happens. B. Pancytosis is an increase in all cell lines, also the opposite. D. Myelodysplasia is a group of cancer where immature blood cells in the bone marrow don't mature properly. While it can cause cytopenias, it's a cause, not a characteristic of hypersplenicity itself. That's why the correct answer here is leukopenia. It's a decrease in white blood cells. Moving on to question number 2. Which of the following organs is responsible for the petty process of RBCs? A. Liver, B. Spleen, C. Kidney, D. Lymph nodes. In here, the correct answer is B. Spleen. So the spleen has two main ways it clears red blood cells. One is culling, which is removing entire whole or damaged RBC from circulation. The other, which this question asks about, is pitting. Pitting is a remarkable process where macrophages in the spleen literally plank out or pit inclusions from within the red blood cells, like Hematology bodies or Papilloma bodies, without destroying the entire cell. The repair RBC is then allowed to re-enter circulation. A. Liver also filters blood, but is primarily involved in culling, not the precise pitting functions. Kidney filters blood plasma to make urine, and lymph nodes filters lymph from pathogens. Neither performs the pitting of RBC. Let's look at question 3. Spherocytes differ from normal red cells in all of the following except A. Decreased surface to volume, B. No central pillar, C. Decreased resistance to hypotonic stimuli, D. Increased deformability. This is an except question, so you are looking for the false statement. The correct answer is D. Increased deformability. Let's analyze why. A spherocyte is a red blood cell that has lost its normal biconcave disc shape and has become spherical. Decreased surface to volume. That is true. A sphere is the shape with the least amount of surface area for a given volume. This is the fundamental problem with the spherocyte. No central pillar. This is also true. Normal biconcavity is thinner in the middle, creating that center on a blood smear. Because spherocytes are round, they look uniform dense with no central pillar. C. Decreased resistance to hypotonic stimuli. This is also true. This is the principles of osmotic fragility test. Because they have less surface membrane to expand, they can take or match water in a hypotonic solution before they burst. So the correct answer is increased deformability. This statement is false. Spherocytes have very decreased deformability. their rigid spherical shape makes it very difficult for them to squeeze through the narrow sinusoids of the spleen, which is why they get trapped and destroyed there. Question number 4. Which of the following is not associated with hereditary spherocytosis? A. Increased osmotic fragility. B. MCAC greater than 36%. C. Intravascular hemolysis. D. Extravascular hemolysis. Again, you are looking for the when that is not associated. So the correct answer is C. Intravascular hemolysis. Hereditary spherocytosis is the classic example of extravascular hemolysis. The term extra means outside the blood vessels. The fatty spherocytes are removed from the circulation by macrophages, primarily in the spleen. So, extravascular hemolysis is definitely associated with the condition. So, let's look at the others. Increased osmotic fragility. As we just discussed, this is the hallmark lab test for spherocytosis, so this is associated. MCAC greater than 36%. This is also a classic finding. MCAC is the mean corpuscular hemoglobin concentration. Because the cell membrane is lost, but the hemoglobin inside is not, the concentration of hemoglobin within the smaller cell volume is increased. It's one of the few conditions where you will see a truly elevated MCAC. So the last way, intravascular hemolysis, is the one that is not associated. Intravascular hemolysis means the red cells are bursting within the blood vessels. This is seen in conditions like PNH, TTP, or severe transfusion reaction, but not typically in hereditary spherocytosis. Last question for today. Which of the following disorder has an increased osmotic fragility? A. Iron deficiency anemia. B. Hereditary elliptocytosis. C. Hereditary stomatocytosis. D. Hereditary spherocytosis. The answer is, of course, D. Hereditary spherocytosis. As we've established, hereditary spherocytosis is the poster child for increased osmotic fragility due to that low surface area to volume ratio. Let's quickly look at why the others are wrong. Iron deficiency anemia. These cells are microcytic and hypochromic, often appearing as thin target cells. They have a high surface area to volume ratio and are actually more resistant to osmotic lysis, so they have decreased osmotic fragility. So B and C, hereditary elliptocytosis and stomatocytosis. Why these are also red cell membrane defects. Their defects on osmotic fragility is very well and not as pronounced or consistent as it is in spherocytosis. Typically, it is normal or only slightly increased. So, when an exam asks the classic disorder with increased osmotic fragility, the answer is almost always hereditary spherocytosis. This is for our first practice session. Thanks for starting with me and see you in the next video.